right. Now is as good a time as any. So my name is William, one of the second-year psychiatry residents here. I'm really excited to have Charles Nichols here tonight to speak with us. And I'll turn things over to Azim to do the introduction.
All right, thanks so much. And thank you so much, Dr. Nichols, for being here. So Dr. Nichols is going to talk to us about the anti-inflammatory properties of psychedelics and these antidepressant-like effects in preclinical models. And just to introduce Dr. Nichols, he earned his BS at Purdue University and his PhD at Carnegie Mellon, studying the developmental neurogenetics in Drosophila melanogaster, and performed his postdoctoral work at Vanderbilt University School of Medicine in the Department of Pharmacology, where he was researching mammalian serotonin 5-HT2 receptor neuropharmacology. His research focused on 5-HT2A receptors and psychedelics for the past 24 years, where he's made several key discoveries. These include the identification of populations of cell types within the brain that directly respond to psychedelics, the development of animal models to study the antidepressant effects of psilocybin, and the discovery of potent anti-inflammatory effects of psychedelics. He's currently a professor in the Department of Pharmacology and Experimental Therapeutics at LSU Health Sciences Center in New Orleans. And before we get started, I'd like to remind everybody that we'll be having questions at the end of this. We'll be moderating that. You can either type your questions into the chat or use the raise hand button, and we will call on you and you can ask your question at that time. William, I think I did that housekeeping part correctly, but if there's anything I missed, please jump in.
No, that's perfect. Yeah. After
Wonderful.
the lecture, yeah, we'll just use the raise hand button like normal. And we usually give preference to people who are willing to talk. So like seeing your faces on the screen makes for a better conversation. So
yep, perfect. I like that. All right. So without further ado, Dr. Nichols, the floor is yours.
All right. Thank you for inviting me to speak to you today in the introduction. Talk to you a little bit about my research down here in New Orleans. First, to get this out of the way, I have to disclose that I'm on the scientific advisory board of Elucis Therapeutics. I have a sponsored research contract with Elucis Therapeutics, and I'm also a venture partner with Palo Santo Ventures. So that being said, I would like to start with serotonin, which is a small molecule hormone and neurotransmitter otherwise known as 5-hydroxytryptamine. And serotonin itself is very evolutionarily ancient. Serotonin and its receptors are found in single cell organisms like Dictyostelium, Paramecium. They're in worms, they're in higher organisms like clams specifically hold an important role in the history of serotonin, in that it was a heart rate prep from clams that was used to identify that there was serotonin in mammalian brain back in 1952 by Betty Tworog. And serotonin is found in higher organisms like insects and mammals. In mammals, there are 14 different serotonin receptor subtypes that are in 7 different families. And because it's been around for so long, serotonin is involved in nearly every physiological or behavioral or developmental process that you can really think of in some form or another through these receptors. So it's very ubiquitous through nature in animals. It also is very important in plants. It's found in several of the plants probably in your gardens, spinach, tomatoes, onions, peas, it's in bananas, it's in pineapples. The role of serotonin in plants, one of the primary roles is as an antioxidant in plants, but it's also necessary to regulate more sophisticated processes like regulation of growth, flowering of plants. And the mechanism of this isn't really quite known, but there's mounting evidence that serotonin is a ligand for auxin binding protein to mediate some of these hormonal effects in plants. In mammals, serotonin is synthesized from tryptophan through this intermediate 5-hydroxytryptophan, Whereas in plants, tryptamine is the intermediate between tryptophan and serotonin. Furthermore, in plants, serotonin is catabolized to several CNS active substances, including CNS active tryptamines like 5-methoxy N-N-dimethyltryptamine, or DMT, found in Psychotria viridis, which is one of the active constituents in the ayahuasca. And as in psilocybin, which is the tryptamine prodrug of psilocin, which is found in several species of mushrooms. And the role of these catabolites of serotonin in plants is not known. Maybe they have similar roles to serotonin. There's also other psychedelics that are naturally found in plants, such as those of the phenethylamine class, for example, in the peyote cactus, and ergots, like ergotamine, found in Claviceps purpurea, which is a fungus that grows on rye. So there are a lot of different molecules in nature that either are involved in the formation of serotonin or derive from serotonin, that in plants, we really don't know what role they play, but humans discovered the psychoactive effects of several of these compounds found in nature millennia ago. For example, morning glory seeds contain ergots. Mushrooms contain psilocybin. On the right is an Aztec statue of the god Xochipilli, depicting morning glories and mushrooms along with tobacco and nicotine. And there is one school of thought that has proposed that the Eleusinian mysteries in ancient Greece evolved some form of either ergot or mushroom to produce an intoxicating effect. The first synthetic psychedelic was lysergic acid diethylamide that was discovered by the Swiss chemist Albert Hofmann in 1938. And at that time, he didn't really notice anything unusual or special about this molecule, and he essentially shelved it as part of the program he was working on at the time. 5 years later, he had some inkling that he missed something about one of these particular compounds that he had synthesized in 1938. So in April of 1943, he went back into the lab, went back to this particular compound and resynthesized a batch of this, lysergic acid diethylamide, number 25 in the series, and started having some strange effects at the end of the day. And he thought it must have been due to this LSD that he was working with. So 5 days later, he decided to give himself basically ingest a small amount of LSD. And at that point, he took, I think it was 0.2 milligrams of LSD. And drugs weren't very potent at that time. So he thought that would be maybe a very low dose. But what we now know is that 0.2 milligrams of LSD is a fairly large, hefty dose. So on April 19th, 1943, he ingested 0.2 milligrams of LSD, started succumbing to the behavioral effects, and had to be helped home on his bicycle by his assistant. And there was a very detailed description of this trip in a book that he wrote called LSD, My Problem Child. Serotonin was discovered in 1949 by two different groups. One group called it antiramine, another group called it serotonin. And in 1952, as I mentioned, it was found in mammalian brain by Betty Tworog. And at that time, the psychoactive properties of LSD were known, and the structure of serotonin was elucidated. It was hypothesized by Woolley and Shaw in 1954 that the ability of LSD to induce a psychomimetic state, along with the structural similarity to serotonin, the hypothesis formulated was that mental illness actually had some biochemical basis, some maybe deficiency of serotonin, and that mental illness like schizophrenia or psychosis was not really Freudian in nature. And really, this is one of the defining moments at the dawn of modern psychopharmacology. The psychedelics we now know are all agonists at the serotonin 5-HT2A receptor. This is a map of the 5-HT2A receptor expression in the human brain. And there's high expression in several areas, with the highest expression in cortical areas like the frontal cortex. And we know that engagement of serotonin 2A receptors is necessary and sufficient to produce behavioral effects both in animals and in humans. So we now know that the psychedelics behavioral effects are induced by activating serotonin 2A receptors in the brain. Several years ago, PET scans in healthy volunteers identified areas of high metabolic activity, for example, in frontal cortex areas. Shortly after that, fMRI scans using BOLD identified altered blood flow in the brain following psychedelic administration. And more recently, high resolution fMRI scans have looked at network connectivity, both in the baseline state compared to the psychedelic state. And what I'm showing here now is some data from Robin Carhart-Harris's group at Imperial College in London, looking at the default mode network connectivity map. And in this circle, each of these smaller circles around the periphery represents a brain area, and the line connecting them represents the strength of the connection between different brain areas. And this is the default mode network, or the connectivity map of the brain when it's not really doing anything. It's sort of in a zoned out mode. However, Robin found that in the brains of individuals after being given psilocybin, that there was this disintegration of the default mode network followed by this massive hyperconnectivity between brain regions. There are brain regions that have reduced connectivity, but by and large, there's a net increase in connectivity, and you have areas of the brain talking to one another that would not normally be talking to one another. So we now have an understanding of how psychedelics alter communication between brain networks, specifically disintegration of the default mode network and increased functional connectivity. And there have been several imaging studies. There have been network-based studies, clinical trials, for example, looking at the use of psilocybin or other psychedelics for substance use disorder, for depression, for anxiety, and to map basic circuits underlying normal cognitive function. But these imaging and clinical studies are really only part of the picture. They don't really depend on any particular knowledge of molecular mechanisms. And by and large, they're largely observational and inform on fundamental brain network function by telling us what brain networks underlie specific aspects of normal consciousness and perception, using psychedelics as tools to alter normal network function in specific ways that are relevant to understanding certain cognitive processes relative to understanding brain network connectivity and function. But these imaging or clinical studies inform on how psychedelics, they don't inform on how psychedelics are interacting with their target receptors, how they're affecting the cells within the brain to produce these changes in network connectivity. And understanding these molecular and cell biological mechanisms is really critical for further developing psychedelics as therapies across several areas of biology, not just for psychiatric disorders. And this is where my research has been focused over the last 24 years or so, is understanding how psychedelics interact with their target receptors to influence effector pathways and cellular function within the brain. So now we'll go to the brain. There's a high expression and density of serotonin 2A receptors in cortical areas with enrichment in layer 5 of the cortex. And this is an fluorescent antibody stain for serotonin 2A receptors. And now we've switched to the rat brain. This is an image of a rat, one of the former rats from my lab, medial prefrontal cortex. There's a good staining density in layer 5. Most of the receptors express on the cell bodies and the apical dendrites, the highest expression. And if we look further in at these receptors, the Golgi stain image from our lab at the medial prefrontal cortex, within the extracellular membrane, the serotonin 2A receptor itself is a G protein coupled 7 transmembrane spanning protein. This is a ribbon cartoon map of the 5-HT2A receptor with LSD in the active binding site. And the crystal structure of LSD in the 5-HT2A receptor was published very recently by Brian Roth's group, and my father was a co-author on this paper. And one of the interesting things about this receptor and LSD's interaction with the receptor was extracellular loop 2. They found that when LSD is in the receptor, but not other types of psychedelics, like for example, a phenethylamine psychedelic, this extracellular loop 2 folds down over the receptor and traps LSD and other ergolines in the receptor and keeps it signaling for very long. So even though LSD is a weak partial agonist at the receptor for canonical signaling, it's a very, very potent psychedelic. So the theory now is that LSD is so potent a psychedelic because, unlike others, it's getting trapped in the receptor and signaling for several hours. Another important aspect of the serotonin 2A receptor is functional selectivity at the receptor. And it's been shown by several groups that different drugs interacting with the receptor can activate and recruit different signaling pathways. For example, serotonin, which is the natural ligand for this receptor, the canonical pathway that it recruits to the receptor is the G-alpha-Q pathway, which activates phospholipase C beta, resulting in intracellular calcium mobilization. There's some evidence that psychedelics, in addition to G-alpha-Q, can recruit additional pathways, perhaps G-alpha-I, to activate phospholipase A2 and arachidonic acid. And then in my lab, we've discovered that psychedelics activate as yet undiscovered pathways that have anti-inflammatory effects within cells. So now we'll go back to the brain again, where serotonin 2A is expressed. We can look at which cells have become directly activated by psychedelics by using a fluorescent antibody for the protein C-FOS, which is an immediate early gene and an indicator of cells that have become transcriptionally active and have likely also become electrically active. Now you can see there's a good overlay between activation of cells and where the 5-HT2A receptor is expressed. Several years ago, when I was a postdoc at Vanderbilt in the Sanders Bush lab, as well as in Ariel Deutsch's lab, and then we've recently confirmed this, it was found that something that was kind of confusing at the time, where this is an image from my laboratory of cells within the prefrontal cortex, showing where the serotonin 2A receptor expressing neurons are. And you can see here outlined in orange, these are the cell bodies of serotonin 2A receptor expressing neurons in the brain of a rat that was given a high dose of a psychedelic drug called DOI, which is a selective agonist for 5-HT2 receptors. And what we noticed was that the cells that became activated were not the cells that predominantly expressed serotonin 2A receptors. You can see here the C-FOS cells that become activated are outlined in these green triangles. So it wasn't really known why the cells that were becoming activated weren't the cells that had high levels of receptor expression. But if you look close enough, you can see that there is a small number of cells that express the receptor that do become activated. So we set out several years ago to identify and purify these cells, find out what was different about them versus the cells that don't become activated. And my hypothesis at the time was that this subpopulation of serotonin 2A receptor expressing neurons that becomes directly activated by psychedelics represents a uniquely responsive subset of neurons in the cortex, and that these cells serve as the trigger for the overall effects that produce the subjective psychedelic trip. And further, that they play a role in modulating normal cognitive processes, and I called this ensemble of cells the trigger population. And we developed new flow cytometry methodology that we call neurocytometry that allows us to isolate cells from fresh, frozen, and fixed brain, routinely fixed human postmortem brain. We don't just isolate bare nuclei. As you can see in this sorted cell micrograph over here, we have cell processes, we have cytoplasmic membrane, we have cytoplasm. So we don't need genetically tagged animals. or cells. We can use antibodies and epitopes for cytoplasmic, as well as extracellular membrane proteins, and we can even sort cells based upon serotonin 2A receptor expression itself. We published on this a few years ago in ACS Chemical Neuroscience if you want to know more about this technique. But using this technique, we were able to sort out and identify the trigger population of cells, do gene expression analysis, and look at what the nature of these cells were. I'm not really going to get into too much detail. I'll give you the summary of that study here in this slide. If you want to know more about this, I'll refer you to this paper. But in general, we found that there are about 5% of cells within the brain in the different cortical regions that become directly activated by psychedelics. We use LSD as well as DOI in these studies. And the neurons that become activated have about 4 to 5-fold higher levels of expression of serotonin 2A receptor mRNA. So they likely express more receptor and are thus more sensitive to both serotonin and psychedelics. We found that there are regional differences in the nature of how these cells become activated. For example, when we're looking at the mGluR2 receptor expression, we found that only neurons in the somatosensory cortex, but not the medial prefrontal cortex in the activated population, were enriched with mGluR2 receptors. We also found that there were inhibitory GABAergic interneurons that were activated. And of these, only 2 subtypes were activated, the parvalbumin and the somatostatin. And how they were activated was regionally dependent. For example, in medial prefrontal cortex and somatosensory cortex, parvalbumin interneurons were activated. Somatostatin interneurons, however, were only activated in the somatosensory cortex. We also found that there were glia and astrocytes that became activated. So you can't really think of psychedelics as activating a specific brain region or another, as in, activating medial prefrontal cortex, activating the temporal cortex. That psychedelics are activating subsets of excitatory, inhibitory, and non-neuronal cells in these complex combinations that differ from one brain region to another. And I think it's this heterogeneity of how these cells are becoming activated in individual brain regions that underlie the heterogeneity of the different network connectivity changes that psychedelics produce, as seen by the imaging studies. So my model of what I think psychedelics are doing at the cellular level, I'm trying to represent in this graphic here, where if you have in layer 5, you have pyramidal cells, and you have a subpopulation of pyramidal cells that are expressing a higher level of 5-HT2A receptors that are more sensitive to both serotonin and to psychedelics. We have glutamatergic inputs and serotonergic inputs from the raphe that normally keep these cells in homeostasis. You also have somatostatin interneurons that mediate slow oscillatory waves. You have parvalbumin fast spiking interneurons that mediate gamma oscillations. And all of these cell types express serotonin 2A receptors. But when you have a psychedelic on board, you are selectively activating a small subset of neurons to enhance glutamatergic transmission, but you're also interfering with the function of somatostatin interneurons and parvalbumin interneurons that I think leads to a local desynchronization of cells within any given brain circuit. And this is happening throughout the brain slightly differently depending upon the brain region. And this is what I think is really cellularly and molecularly underlying the disintegration of the default mode network that is seen in the imaging studies. But what are the consequences of all these effects, aside from disintegration of the default mode network, hyperconnectivity? Well, one of the consequences are that psychedelics have been found to have antidepressants and anxiolytic effects in humans as a result of potentially these biochemical and cellular changes. This is some data from the Griffiths group at Johns Hopkins and a 2016 paper showing that for depression and anxiety after a single dose of psilocybin, that there is a significant and profound reduction of depression that is long lasting. In this study, it was out to 6 months. after a single dose of psilocybin. More recently, there was a follow-up study from Steve Ross's group at NYU looking back at his data sets, showing that 3 to 4 years later, 70% of these patients treated with a single dose of psilocybin had not had a remission into depression. So a single treatment of psilocybin really has these long-lasting effects of not only antidepressant activity, but also in substance use disorder. There have been profound effects shown with alcohol abuse, nicotine use, and in fact, the FDA has designated the study of psilocybin with breakthrough status for phase 3 clinical trials for the development of psilocybin for the treatment of major depression. So there are still several questions at this point. How does administration of a drug lead to these persistent therapeutic effects? These clinical studies really don't get at mechanism. And within this, are the antidepressant effects or effects against substance abuse dependent upon the subjective peak experience or the trip that the individual experiences? And I think to answer these questions, we really needed to develop animal experimental systems. And so we set out several years ago to develop model systems that we could study some of these molecular and genetic questions to identify what's going on in the brains that leads to these long-lasting persistent therapeutic effects. And we developed 2 rodent models. One is using the Wistar Kyoto strain of rat. And this is a strain of rat that is considered an endogenous model of depression. It's a depressed rat from genetically. They act depressed, they respond to antidepressants. And another model that we developed was using normal Sprague-Dawley female rats in an adolescent chronic restraint stress paradigm that was developed by the postdoc in my lab, Dr. Megan Hibicki, when she was a graduate student at Mercer. The nice feature about this model is it's stress-based and it elicits lifelong behavioral deficits that are relevant to depressant-like behaviors in these rats, and it's effective in females, which several models of stress and depression aren't effective in females. So in these models, we would administer a single dose of psilocybin, and then 5 or 6 weeks later, we would test in several outcome measures like the forced swim test, object pattern separation, and elevated plus to look at therapeutic efficacy. So with the WKY model shown here, normally a WKY rat will act depressed. You put it in a forced swim tank, it just floats there. It has high immobility, and this is a model of helplessness. When we take these rats, we give them a single dose of psilocybin, and then 5 weeks later, we put them in the FST tank. This is how they behave. They have significantly reduced immobility, increased swimming, and increased climbing. And in fact, they behave virtually identical to a normal Sprague-Dawley rat in this assay. You could read more about this study. It was published about a year ago in ACS Chemical Neuroscience, where we also compared it directly to ketamine. And in this model system, ketamine had transient antidepressant-like effects lasting about 2 weeks, very similar to what's seen in humans. And we also looked at LSD, and LSD was similar to psilocybin, and a single treatment had long-lasting persistence effects out to the 6 weeks, five or 6 weeks that we tested. So the next model I'll tell you about is the Adolescent Chronic Restraint Stress Model, or ACRS. As I mentioned, this was developed by Dr. Hibicki and is effective in females and is stress-based. So therefore, it has a much higher translational relevance to the human demographic than, say, something like the WKY strain in the forced swim test as the outcome measure. This model, during adolescence, for 1 hour a day, we put these rats in essentially a frosting bag for 1 hour. And then 7 days after the last restraint stress, we'll give a single treatment IP injection of psilocybin in young adulthood. We wait 5 weeks, and then we do an object pattern separation test and this is a test that is cognitive-based, that I'll talk about in the next slide. This paradigm produces lifelong depression-like symptoms in these female rats and has been validated. Traditional antidepressant medications are effective in normalizing these rats and their behavior. And the object pattern separation is a task that has no neural circuitry, the entorhinal cortex, the dentate gyrus, the CA3 of the hippocampus. The object pattern separation is also a behavior that is found in humans that is mediated by the same circuit, and there are tests in humans that can look for this. And pattern recognition and separation is defective in several different psychiatric disorders, including depression. So we have a translationally relevant model here with an outcome measure with defined neural circuitry that's conserved between rats and humans. So the test essentially looks at two identical objects within a circular arena. And the first day, they're in the same plane. On day 2, they're moved out of the plane. On day 3, a little bit more. Day 4, day 5, move further out of the plane. And it's measuring the ability of this animal to notice when this pattern has changed, when the position of one identical object has moved with respect to the location of another object. The data that I'm showing here are on the right of non-stressed animals, and the discrimination index is the ability of the animal to discriminate this moved position. And it's showing from day 5, which is the farthest out position. These animals have very good discrimination, both the control animals and the animals that were just treated with psilocybin. So psilocybin really has no effect. In the stressed animals, they really display no ability to discriminate this pattern separation. However, in the stressed animals that received a single treatment with psilocybin, 5 weeks prior to this test, we have normalized this behavior. So we've normalized the dysfunction within this hippocampal circuit so that it is now able to recognize pattern separation again. So combining the stress-based depression-inducing model with this cognitive-based translational pattern separation outcome measure, I think we have a very good system to study the long-lasting effects of psilocybin and what it's doing in the brain. Another model that we've developed is in Drosophila using fruit flies. And we have essentially found that psilocybin has antidepressant-like effects in fruit flies. And for this, we adapted a forced swim test that was initially developed by Wendy Neckemeyer at St. Louis University and validated it. And it's the same principle as you would have for a rodent forced swim test. You put flies in a liquid and measure their immobility over a period of time. And we tested citalopram, which you know is an SSRI that's commonly used to treat depression. And we have identified a substrain of a wild type fly that essentially acts depressed. It performs poorly. It has high immobility in the forced swim test compared to another wild type strain that has very high mobility. So with this strain of fly, if we treat this fly with citalopram for 5 days with citalopram in the food, we can decrease the immobility in this fly down closer towards wild type levels. If we just feed this fly citalopram for 1 day and then test the fly 5 days later, we see this has no effect on the immobility. So this is similar to what's seen in humans, for example, is that an SSRI has to be taken chronically for it to have therapeutic benefit. We're seeing something similar in the fly. If we give the fly a high dose of psilocybin in the food, feed it for 1 day, or even a relatively low concentration of psilocybin in the food for 1 day, and then test 5 days later, we see an antidepressant-like effect nearly equivalent to that of the chronic citalopram. So here we have a potential genetic model in fruit flies where psilocybin is having a long-term antidepressant-like effect. And this provides an additional genetic model that we can now look at some of the fundamental neurochemistry underlying these effects of psilocybin on circuits relevant to depression and depression-like behaviors. So summary of psychedelics to treat depression so far. Antidepressant-like effects are long-lasting in rats and in flies. We found that the environment the rats experience in the days following drug treatment shapes outcome. And the psychedelics open a window of behavioral flexibility that allows the animals to develop better coping skills. And this is data that I didn't show, but is in the ACS paper that I mentioned. And we recapitulated results found in clinical studies of the long-lasting effects. And importantly, this provides experimental systems to collect tissues from for analysis. And overall, we believe that the antidepressant effects in humans are likely biological in nature, and that while correlated to antidepressant effect, that the peak ego dissolution and the subjective experiences are not causational to the antidepressant effect. But that doesn't mean that they're irrelevant. I think there's an interplay with the subjective experiences and the post-experience integrative therapy that's really necessary to maximize the therapeutic efficacy. And currently we are analyzing the brains of these rats, looking at the genetic gene expression, proteomics, and epigenetics to see if we can identify what the molecular mechanisms are. So what I'd like to do now is dramatically switch gears to another aspect of psychedelic pharmacology. The serotonin 2A receptor is the most widely expressed receptor throughout the body. It's in the cardiovascular system, the GI, the bone, muscle, skin, eyes, and at the 5-HT2A receptor, serotonin is primarily pro-inflammatory. And we discovered several years ago that psychedelics acting at this receptor are anti-inflammatory. They have the opposite effect of serotonin. And it's not merely blockade of the pro-inflammatory effects of serotonin, that we're actively inducing anti-inflammatory mechanisms. One of the models that we've looked at is a model of vascular inflammation, a ApoE knockout mouse that develops vascular inflammation, metabolic dysfunction, and atherosclerosis after several weeks of feeding on a high-fat, high-cholesterol Western diet. And in this model, we took these mice and implanted osmotic mini pumps that slowly release this psychedelic drug DOI into systemic circulation during the 16 weeks of high-fat feeding. There were no behavioral or weight observable or overt differences in the control animals that had saline in the pumps versus the ones that had DOI in the pumps. And we took out the aortic arch from the hearts of these mice and looked at pro-inflammatory biomarker expression. Example of some of those is shown here. In the normal chow-fed mice, IL-6 expression is shown. IL-6 is a key pro-inflammatory cytokine. The DOI itself had no effect. The high-fat fed animals, there's a significant increase in the expression of IL-6. This indicates vascular inflammation. However, the animals fed a high-fat diet that were also receiving DOI during this time really had no vascular inflammation that was evident. And we see this with IL-6. We saw this with another pro-inflammatory biomarker, VCAM1, these really astounding anti-inflammatory effects. The animals fed DOI also had a reduction in the high-fat induced cholesterol levels, and DOI improved glucose tolerance in these mice. So we did a glucose tolerance test, inject glucose, and then measure blood glucose level over 2 hours, where it peaks and then comes back down. The high-fat fed group is shown in the pink trace, and the high-fat fed DOI group in the trace below that. So you can see that there's better glucose tolerance in the group that was treated with DOI. So DOI does not just have anti-inflammatory effects in this model of cardiovascular disease, but is also having protective effects against metabolic disease and diabetes. The primary model that we've used to look at anti-inflammatory effects has been allergic asthma. And asthma is a pulmonary disease characterized by bronchial inflammation, eosinophilia, mucus overproduction, and airways resistance that lead to difficulty in breathing. And asthma is a nice model to study because it involves several aspects of the immune function that are all acting in concert. It brings in innate cell immunity with macrophages, adaptive cell immunity with T cells, other cell types like endothelial cells, muscle cells that are also involved in immune processes, mast cells. So it's really like a perfect storm, so to speak, of immune cell function that we can look at several aspects of the immune system with this. Current therapies include beta blockers, which are not disease modifying, and immunosuppressants like corticosteroids. But there's a substantial population of severe asthmatics that don't respond to either of these types of therapies. And the general gist of our asthma model is to generate mice with asthma and treat that asthma with a psychedelic administered by an inhaler. One of our primary protocols is a prophylactic protocol that's shown here, where we take chicken egg white ovalbumin and administer it on day 0 and then day 7 intraperitoneally to prime the immune system to respond to this allergen with an allergic reaction. And then I've shown here in this particular scheme on day 14, 15, and 16, they're exposed for 20 minutes to nebulized ovalbumin to start the immune reaction in the lung, which peaks at 48 hours later in the symptoms that are highly correlative with asthma. And we measure pulmonary function in the awake, freely moving animal in whole body plethysmography chambers. And we can look at airways resistance using whole body plethysmography. And this is a good proxy assay for inflammation. If there's inflammation in the lung, it's going to induce effects which are going to make it difficult for this animal to breathe. So we challenge these animals nebulized nose only with methacholine, which is a muscarinic agonist that produces a mild bronchial constriction. And airways resistance with increasing methacholine goes up a little bit. With the asthmatic animals, there's a hyper-responsiveness, as you see in the red trace. These animals have much more difficulty in breathing at the higher doses of methacholine. So we challenge these animals, we give them the methacholine, and you see in the DOI treated group that these animals have breathing parameters essentially equivalent to the non-asthmatic animals, indicating that there's no pulmonary inflammation within these animals. Another paradigm that we've used is a rescue paradigm in this acute model, where instead of giving the drug prior to the ovalbumin on days 14, 15, and 16, we're giving it 6 hours prior to the plethysmography. And this is around a time point when the symptoms of asthma are peaking. And what you can see here is not a complete rescue, but there's a significant decrease of about 50% in the hyperreactivity or hyperresponsiveness of these animals to methacholine. So we're having an anti-inflammatory effect, even with only a 6-hour pretreatment. And if we look at the T cells within the lungs of these animals, we see a very pronounced anti-inflammatory effect. In a naive control animal, there's a certain baseline number of activated T cells. In the asthmatic ovalbumin treated animal, there's a significant increase in the number of activated T cells in the lungs. DOI itself doesn't have any effect on T cells. In the prophylactic protocol, we see that there's a marked reduction in the number of T cells recruited to the lung. So we're preventing recruitment of T cells to the lung. And in the rescue paradigm, we can see that in only a 6-hour pretreatment, we've reduced the number of activated T cells in the lung from this pathological state essentially down to baseline normal state. So even with this short pretreatment, we're having a very pronounced anti-inflammatory effect within the lungs. We're also blocking recruitment of eosinophils to the lung. So it's not just effects on T cells, it's on other cells like eosinophils. And if we look at the lungs themselves, that's shown in this slide. This is the acute prophylactic model. This is an airway on the top, stained with PAS, which stains mucus pink. H&E stain on the bottom, which is a zoomed out view, you can see that in the asthmatic animals, there is a significant thickening of the airways, there's mucus overproduction, and there's inflammation around the airways. In animals treated with psychedelics, and not just DOI, but several other psychedelics, we completely reverse the mucus overproduction and eliminate inflammation in the lungs. And we've seen this down to a very, very low dose. The EC50 dose for these anti-inflammatory effects in our asthma model is 0.005 milligrams per kilogram. And the drug doesn't have to be given nose only. We can also administer it intraperitoneally with an injection. It has the same potency and efficacy. And this is about 30 to 50 times less than the behavioral potency, the minimal behavioral threshold to induce any psychedelic-like behaviors in these animals. So it's essentially microdosing. But what about reversing chronic asthma? That's more relevant to human disease. So for that, we generated a model in mice that we repeatedly exposed these mice to ovalbumin once a week over 4 months. And this generates symptoms of asthma that are persistent in the animals. In the acute model, the symptoms will go away after about 72 hours. 3 to 4 days later, they're back to baseline. So we needed a model where these symptoms did not return to baseline within a few days. And within this model, we found that the symptoms of asthma persisted at full strength at least 2 weeks after the final ovalbumin exposure. And in this model, we gave a single administration, nose only of DOI once a day for 4 days. And the first treatment of DOI was 10 full days after the final ovalbumin exposure. And then 24 hours after the final DOI exposure, we did whole body plasmagraphy, but we also took the lungs out for pathology. One of the symptoms of asthma in humans that we were able to recapitulate in our model was remodeling of the airways, collagen deposition and fibrosis, which is stained here in green. So you can see that the airways are thickened, there's mucus, but now we also have fibrosis in these lungs. But in the animals that were treated with DOI, we not only see a return of the airway thickness to normal, mucus is gone, but we have a 70% reduction in fibrosis. We've reversed the fibrosis in this lung. This may be due to, we show that we are decreasing TGF-beta expression in the lungs. That may be a part of the mechanism. And in these animals, they can also breathe normally. The DOI treated animals in this plasmathography experiment is not different from naive. So with DOI, we're able to prophylactically prevent inflammation in the development of asthma. We're able to acutely rescue inflammation and symptoms associated with asthma. And we're able to reverse chronic asthma and fibrosis with psychedelics. And our model for this is shown here, where we have T cells that normally express pro-inflammatory cytokines. We have macrophages that also express pro-inflammatory cytokines. B cells leading to mast cells, which degranulate with histamine and leukotrienes. Other pro-inflammatory cytokines like IL-13 lead to goblet cell hyperplasia and mucus overproduction. GM-CSF and IL-5 both lead to recruitment of eosinophils in the lung that are largely responsible for pulmonary damage and the airways hyperresponsiveness and the inflammation. All of these cell types express serotonin 2A receptors. And what we believe is happening is that activation of serotonin 2A receptors with psychedelics, but not serotonin, because it's been shown in the literature by other groups that serotonin has a pro-inflammatory effect on these same systems. But DOI and other psychedelics activating these receptors lead to a selective suppression of certain pro-inflammatory cytokines. So we don't get activation of T cells, recruitment of eosinophils, degranulation of mast cells, leading to normal lung pathology. But we're not acting as an immunosuppressant. There are several pro-inflammatory cytokines like IL-4 that their levels are not changed. So we're not acting like a corticosteroid. So it'd be predicted that certain aspects of the immune system are left intact, that are able to respond to other environmental factors, and there'll be a fewer side effects associated with 5-HT2A receptor therapy than an immunosuppressant like a corticosteroid. So with this asthma model, in the last few minutes, I'll describe what we're doing with it at the practical side of things. We're using it as a drug discovery platform to look at the SAR of different psychedelics to develop new chemical entities for clinical use. And we published on this last fall in ACS Pharmacology and Translational Science, where we looked at 25 different psychedelics in this asthma model. They're shown here, where we looked at their ability to fully block the development of asthma, have really full anti-inflammatory efficacy, partial efficacy that even at relatively high doses, we're only able to partially suppress airways hyperresponsiveness and inflammation. And we found drugs that were not efficacious at all, that even with relatively high levels of drug, we could not really suppress the airways hyperresponsiveness below those of just the asthmatic animals. And we did not observe any correlation with behavioral potency. For example, the drug 2,4-DMA, it's a phenethylamine drug, has full efficacy in this platform, but Alexander Shulgin has reported in humans in PCOL that it does not have any behavioral activity or behavioral effects. On the other side of the spectrum, we have an N-dimethyltryptamine. DMT is a very powerful psychedelic drug, but in our model system, neither DMT or 5-methoxy-DMT have any anti-inflammatory efficacy. And LSD, which is a very potent psychedelic, even at relatively high levels, is only to partially block the inflammation seen in asthma. We did receptor pharmacology studies looking at canonical signaling pathways, and that agrees with these results, and that there is no correlation between canonical signaling pathways for calcium mobilization or beta-arrestin recruitment with anti-inflammatory effects. So we believe that it is a separate pathway from canonical signaling that underlies behavior that's being recruited. The pharmacophore structure is 2CH, which is 2,5-dimethoxyphenethylamine. This is a drug that provides for full anti-inflammatory, anti-asthma efficacy. And we found that we can substitute the 4 position with halogens. We can add an alpha methyl. This has really no detrimental effect on anti-inflammatory efficacy. We can add various aliphatic chains to the 4 position and maintain full efficacy. But if we rigidify the 2,5-dimethoxys, if we rigidify the side chain, if we lose the 5'-methoxy or modified methoxy, we lose efficacy, although not completely. And if we have a very large substitution group at the 4 position, we also lose efficacy. With ergolines, LSD, as I mentioned, a very potent psychedelic drug, is only partially efficacious. Maybe that's due to LSD being a partial agonist at the receptor. To test for that, we tested a related drug called SLAD, which is a full agonist at the receptor with respect to canonical signaling and behaviors. And it was really no different than LSD. And the psychedelic 5-HT2A agonist glyceride has absolutely no efficacy in this model. For tryptamines, psilocin and 4-hydroxy-DIPT were found to be fully efficacious. However, if we lose the 4'-hydroxy to DMT, then we lose all efficacy, and this becomes really a non-anti-inflammatory molecule. So the 4-hydroxy is important. And the nature of the nitrogen substituted groups look like they're can be a methyl or an isopropyl, so we have some wiggle room there. So putting all this data together, we've been designing new chemical entities to have maximal anti-inflammatory efficacy while potentially reducing behavioral liability. And we've developed several NCEs that I partnered with Elucis to develop for the clinic, and they're currently now in drug development to develop some of these more promising ones that we hope to be in clinical trials the next year or so as an anti-inflammatory. And our first indication will be an ocular inflammatory related disease. So summary of anti-inflammatory effects, we have proof of principle data in vascular inflammation models, metabolic dysfunction, inflammatory bowel disease, asthma, ocular inflammation. This is in collaboration with Tim Foster, who's upstairs in the Department of Microbiology, Immunology, and Parasitology. Key features of serotonin 2A agonist therapies, that they're efficacious at sub-behavioural levels. In the case of these fully efficacious drugs like DOI, 30 to 100 times less drug is necessary to produce anti-inflammatory efficacy than minimal behavioral levels. And these drugs are steroid sparing. They're not a general immunosuppressive, so they should have fewer side effects and predict that they may be effective in some of the more severe cases of inflammatory disease where you might not want to use steroid. I'd like to acknowledge the members in my lab that have contributed to this project, specifically Megan Hibicki, who developed the ACRS protocol, and we're currently using this now to explore additional aspects of psilocybin as an antidepressant. Thomas Flanagan, who headed up the Anti-inflammatory Asthma Project. My collaborator, Tim Foster, who's working with me on the anti-inflammatory project and has developed the ocular side of things. And my father, David Nichols. It's been a pleasure to professionally collaborate with him over the last 20 some years, even though he advised me that if I went into psychedelics as a career, that it would be the death of my career. I'm glad to have proved him wrong, whether that's my stubbornness or tenacity, that's up for debate. I'd like to acknowledge for support Elucis Therapeutics, the American Asthma Foundation, the NIH, Betsy Gordon Foundation, and the Hefter Research Institute for funding over the years. So with that, I will open it up for questions. And thank you for your attention.
Thanks a lot, Dr. Nichols. That was great and fascinating and really impressive work all around. So thanks for sharing all of that with us. We'll start the Q&A here.
There we go.
So, okay. So, like we said earlier, so just raise your hands if you have questions. I see some hands coming up now. And we'll just start calling on people. So the first person I see is Melody Hayes.
Hi, Dr. Hayes here. First, thank you so much for an amazing, compelling presentation. My first question is, you said there was something like trip cells, and then you also said that you implied that the antidepressant effects were not dependent on the tripping effect or the ego dissolution effect.
Right.
This is in contrast to Brian Anderson's data out of UCSF, which actually implied that when he gets like doses less than 3 grams, and people didn't see the antidepressant effect. And then he did a crossover study, gave people the full 3 grams, and that's actually when they had the trip experience is when they saw clinical benefit. That's one question. My next question is, you're seeing, your slide beautifully demonstrated, the final slide demonstrated that you titrated like which psychedelics actually have the most benefit on asthma. In your first presentation, you kept saying that, first part of the presentation with antidepressants, you kept saying psychedelics. And I was just wondering if you could also titrate out like which psychedelics have you found in a lab clinical setting that have the most robust response in regards to antidepressant response? Because we know about psilocybin. I don't know if you found things that were more efficacious than psilocybin.
Okay, so I can answer both of those questions, fortunately. The first, with the peak experience not really being necessary, kind of is an interpretation of a rat or a fruit fly not really being able to have an existential sense of self and their relationship to others and their impending doom at some point in their life that they can brood over. That being said, that's open for interpretation of what a rat or a fly actually thinks or feels. But I think an interpretation of our data, and it's not inconsistent with Brian Anderson's data, is that if you think of the psychedelic trip ego experience as being the biomarker that you've had enough drug to do what's necessary to do in the brain to potentially induce these synaptic plasticity changes and open up that window of behavioral flexibility. So in that case, you would see a correlation, but that does not necessarily mean it's causational.
I'll meditate on that. I'll meditate on that.
Okay. And for the second question, it's much more difficult to test these drugs in our depression models because each of these drugs that we test takes several months to do this. But we are actually doing that right now. In the laboratory, we're looking at a couple different psychedelics that have different receptor selectivity to see if there are different effects. Right now, we've looked at psilocybin, we've looked at LSD, and we've looked at ketamine. Ketamine has a very different profile than psilocybin or LSD, and LSD and psilocybin roughly have the same profile. But these experiments that we're doing now will inform us on a couple different drugs. But that obviously is an area, are there drugs that may be less antidepressant than others, and what would those mechanisms be? We really don't know what the molecular mechanism is and how we can optimize that. So hopefully that will be what we can get out of this.
I guess my global question is, which you're kind of getting at, is can you take a lot of pharmacologic companies are looking at how to take away the trip and still have the benefit. And
right.
So, yeah, I have a theory behind that, but that was like my larger question.
So can you have a non-psychedelic psychedelic with antidepressant effects? I think we can have a non-psychedelic psychedelic for anti-inflammatory effects. It's very convincing that we have different effector pathways there, and the new chemicals that we developed, we've maintained anti-inflammatory potency, and we've decreased behavioral effects by 70%. So we're separating there. But for the CNS side of things, I think it's a much more tricky question because I don't see how you can really dramatically affect the brain and produce all this remodeling and synaptic plasticity without having some sort of behavioral effect at that time that would be obvious. And that's something that I'm not yet convinced of, is that you can have a non-psychedelic psychedelic. It might be possible, but at this point in time, I'm not sure that I would say that was possible.
Thank you.
All right, next, George Anderson. Are you able to unmute?
Okay, yeah, thank you. A fascinating talk, and I just wanted to know whether you've checked to see whether you can block the effects, these anti-inflammatory effects of the DOI and these other related compounds that you're finding with 5-HT2A antagonists.
That has been a difficult question because the way that our model works, we have to have something that's essentially water soluble in order to nebulize it. And the repeated dosing and the 5-HT2A antagonists that are currently available really are only soluble in DMSO. And when we've tried them in our assay, we scar the lungs of the animals or we make them sick. So the approach that we've taken instead was to look to see if we can recapitulate the anti-inflammatory effects with selective 5-HT2B and C agonists. And we cannot with those. And we also have the 5-HT2A knockout mouse. And when we look at the effects of DOI in the knockout mouse, we also have no anti-inflammatory effects. In the cell culture models that we've looked at, where we are able to look at the effects of TNF alpha, we are able to block those with the selective 2A agonist, but not 2B and 2C agonists. So altogether, I'm fairly confident that it is a 5-HT2A mediated effect.
Thanks, Dr. Nichols. See Emmanuel Schindler. Let me unmute you here.
Yes, thanks. I wasn't able to unmute myself. Dr. Nichols, for your talk, and I'm sorry to not turn on my video. I'm at home doing laundry, and it's not very professional. My laundry.
No problem.
And I spent the first half of your talk driving home, so I missed your slides, but I was very I was very interested in what you were saying, and I also enjoyed listening to you at the ISRP meeting back in October.
Okay.
My first time in New Orleans, so that was also very nice. I had a couple of questions. In the trigger population of cells, is there something different about the 5-HT2 receptor, either in the density or co-localization of other receptors, or the binding affinity, or intracellular signaling that makes them different from the other 2A receptors?
We haven't done that work yet. A lot of that sort of work, looking at the function of that, was in an R01 grant that was not funded a couple years ago, trying to get more data on that. We just know that in those cells, there's a higher level of expression of the mRNA at this point. We have plans to try to do some neurocytometry to pull these cells out to see if we can find some functional differences, but we haven't actually done that yet. There may be differences, but I can't say one way or the other.
Yeah, I would find that really, really fascinating if there were differences in those in those cells. And my next question is, in the animal models where you gave a single dose of psychedelic and saw lasting effects, were there did you look at the brains afterwards to see if there were lasting changes in either whether it's way or some other receptor, density or affinity or anything?
So we are currently right now analyzing the brains of these animals. We've dissected out 3 brain regions that we're particularly interested in, and we're in the process of doing proteomics, RNA-seq, and some epigenetic analysis, comparing the different treatment groups at the 5 and 6 weeks to see if we can uncover really any sort of molecular differences at that level. But we haven't looked at receptor expression yet or not. We've essentially ground up half the brains for these studies, but the other half of the brain we're sectioning and slicing to do those sorts of immunohistochemistry studies. And that's the dissertation project of one of my graduate students, Hannah Kramer.
Oh, great, lucky student. And I have one last. It's actually more of a comment on an earlier question. So I've been studying psilocybin in different headache disorders here at Yale, and I found that there was, in my small studies, there's no correlation between the acute psychedelic effects and the lasting reduction in the headache burden. So at least in headache does not seem to be, it does not seem to matter whether the person has a very strong trip or not. Of course, headache disorders are a different animal, but and there really aren't really good models, unfortunately, for headache disease. So I'm always looking for a way to investigate that a bit further. If you have any ideas, let me know.
Oh yeah, it's, I guess even one question, is it really the 5-HT2 receptors there, or is it a combination of the 2 and the 1B or something like that they might be hitting a functionally selective differential activation of the 1D receptor? It's a really fascinating topic. But yeah, very difficult to study.
Yeah, very much so. Well, thanks again.
Benoit, you're up next.
Thank you. Thank you, Dr. Nichols. This was an amazing presentation. The scope of your work is truly vast, and
thank you.
Honestly, still wrapping a lot of my head around it. I personally believe for a long time that the expansion of psychedelics into somatic medicine is really the forefront of the science, and it's my mind is blown away, the depth and thoroughness of your research in that direction. So I'm very, very impressed. I'm curious, this came up in my head when I was thinking about the first half of your presentation. I feel like part of the difficulty with psychedelics in humans and in human clinical application is that they're not able to be assumed to be antidepressant or assumed to be efficacious as treatments. The clinical data is produced in a very specific environment. And it kind of had me questioning or even formulating this challenge as to whether it's possible to develop an animal model of a bad psychedelic experience, because I think that also has a lot of clinical relevance. And I'm wondering if that's something you've ever thought about as well.
Oh, like a bad trip model. Yeah, I don't know. You know, when we give like a single acute dose to the animals, they're not stressed. They're in their cage. They're grooming each other. They're eating. They're sleeping. There's no visible signs of distress. It would be interesting, as I alluded to, the nature of the subjective experience shapes the outcome of the antidepressant effect. And one of the things that we've noticed is that if we take the animals a week after they've been given the psilocybin and we dunk them in the forced swim test tank, rather than just let them sit in their cage, what that does is that reduces the effect size of the antidepressants effect, but it confers a very profound anxiolytic effect on there. So depending upon what you do to these animals, maybe if you shock them or stress them immediately before or after, it would, I think, have an effect on the antidepressant effect. But we haven't really explored that beyond just doing a forced swim test a week after and showing that there was a pretty profound difference in the outcome. But I don't think anybody is really, I don't know how. Yeah, it's a good question. It's a good question.
Yeah, I was just curious. Thank you.
All right, George Greer.
Yes, so to kind of put you on the spot, if someone said they wanted to microdose 100 micrograms of LSD or 100 milligrams of psilocybin mushrooms every 3 days, would you advise them against it because of valvulopathy, or want to expand on that?
The valvulopathy issue is really it's activation of serotonin 2B over time. And there's some recent data that Brian Roth has presented that psilocybin does not activate the 2B receptor as efficiently as LSD does. So there might be less valvulopathy risk with psilocybin than LSD in that case. Not that I think there would be a significant risk to begin with because the dose is so low, but based on my anti-inflammatory work, theoretically, I would go for the psilocybin because psilocybin is a potent anti-inflammatory in our model systems, whereas LSD is it's one of the weakest anti-inflammatories that we've found. So just based on anti-inflammatory, microdosing might be more efficacious with psilocybin on that aspect of things. And from some of Brian's latest data, probably also has less valvulopathy risk associated with it. But I'm not a medical doctor. I can't give medical advice. That's just a theory. Thanks, George.
Thank you. Yes. Yeah. Not a good idea to give medical advice on a Zoom call.
Yeah. So that was just pure speculation on my part.
Thank you.
All right, next, Scott Engel.
Yeah, hi, thanks for the talk. My camera's broken, so I can't zoom in. But I wanted to ask, could you give a brief history of the use of the word tryptamine? You know, its Greek and Latin roots. When was it? When did it first begin to be used? That exact word tryptamine. How long has it been in use?
Oh, a long time ago. It was in use. Oh, at least 100 years, I think. As a not really as a psychedelic, but just to describe what a chemical scaffold is from a chemical standpoint. Yeah, I don't know the history of the use of tryptamine, but it is essentially, it's like an acetoxy or a methoxy. It's a descriptive chemical term, and it originally was used as that prior to sort of being co-opted to define a class of psychedelics.
And yeah, just the word itself is kind of suggestive, and it's just it's curious to me that, you know, in the 60s, the classic description of it taking a psychedelic was to trip, and everybody was tripping, you know?
Yeah.
And now the word tryptamine is used all throughout psychedelic neuroscience.
Oh, true. But the term tryptamine, in relation to its description of, say, of DMT, psilocin, serotonin was definitely in use in the chemical literature prior to people tripping on it. But beyond that, I don't really know much more.
All right, thank you.
I've got a follow-up, sort of different question though. So most of the clinical research that's been done thus far is focused on psychiatric disease, it seems. But so you've got some compelling work with anti-inflammatory issues, and I was wondering what you see as sort of the timeline to potentially bring maybe the first or a handful of anti-inflammatory psychedelic drugs to market where patients are actually getting them.
We would hope to see something on the market in the next 3 to 5 years. We have recently selected lead candidates and indications for some of our lead molecules, and we're doing the IND enabling studies with the FDA. And hopefully this time next year, we'll be doing our first phase 1 safety trial on some of these candidates. We're doing scale up, synthesis, stability, GMP. So assuming that we don't really run into any obstacles between now and then, we hope that this time next year we'll have phase 1 safety data, and then assuming that looks good, proceed. Our first indication is for ocular inflammation in an eye drop, and a couple reasons for that. My collaborator Tim Foster has found some really impressive anti-inflammatory effects in the retina, in the eye in general, and the second reason being that an eye drop is going to have a lower risk of systemic exposure to behavioral effects, and so that'll be lowering the risk of any psychedelic effects with that. So hopefully by the end of next year, we'll have some efficacy data in this, and then in 3 to 5 years, hopefully something on the market. But it's a traditional, basically a traditional drug discovery campaign, like any other kind of anti-inflammatory.
William, I'll jump in with a question as well. So the traditional sort of microdosing schedule has been some number of days off and some days on. I think most of the models that you've shown have been more of a continuous infusion. Just 2 questions around that. So one, can you speak a little bit to this theory of receptor desensitization or sensitization, desensitization, the potential like theoretical underpinnings of this cyclical dosing as it relates to the sort of receptor internalization or downstream signaling? And the sort of second question is, you mentioned that the signaling pathways that are responsible for this anti-inflammatory effect. What I find fascinating is that, it's saying it's 30 to 50 or 100 times lower doses than than the doses for any behavioral effects. So does that indicate that these peripheral inflammatory cells just have different sensitivity to the EC50 on the receptor for that signaling pathway is just much lower? So they're much more potent? Or is there some other interesting like trapping effect that's happening? What's the nature of that effect? And then what is that signaling pathway that's responsible for the anti-inflammatory effects if it's one that's totally different from the canonical ones?
I'll answer your second question first. That's really a main focus of the research in my laboratory right now, and several efforts, is to really identify what this effector pathway is, what the mechanism is. We're beginning to understand now its effects on several aspects of cellular physiology. We've identified some of the molecules involved in this, and it's turning out to be an interesting but complicated story. It's not as simple as we're just activating this one pathway and that's producing the effect. In terms of the sensitivity, I think it's just that whatever that pathway is, that the psychedelics are stabilizing the receptor in the particular conformation that's very efficient at recruiting those effectors to the receptor that serotonin just is not doing. And so we're trying to identify those effectors as well. That's a project of one of my graduate students in the laboratory. So the Asthma Project, what we're doing now is not so much looking at asthma per se, but we're using that as a platform to harvest different cells of the immune system to see how we're affecting them and do analysis of those to see what are the changes, can we identify the effectors and genes and transcription factors being affected. So that's really a very active area in my laboratory and something I'm keen to understand, because it is such an unusual phenomenon. Again, in our tissue culture, cell culture, the EC50s are 20 picomolar to block TNF alpha, whereas for calcium mobilization, you're in the nanomolar range. So it's something that we were able to translate from in vitro to in vivo, showing this super potency. We don't know what that is. It's really kind of bizarre if you think about it, but what this pathway is. We have some clues, and I think we're on the right trail, but we're actively looking at that. And for your first question on sort of the internalization microdosing schedule, it's the phenomenon of tachyphylaxis is there. Like if somebody takes LSD, it's been shown repeatedly over the course of 2 or 3 days, they lose sensitivity and there's no longer any behavioral effects. And they're shown in animal models, cell models, that there's rapid internalization of the receptor, there's desensitization of the receptor. So if somebody were to take a psychedelic or 2A agonists on a daily basis, the theory is that that's going to reduce the amount of receptors available for activation, desensitize these receptors, and you're effectively going to be titrating away the therapeutic mechanism. So by waiting 3 or 4 days, you're allowing these receptors to resensitize back to the signal. And in work that my father has done in some of his rat models, he's shown that if you give LSD to a rat, not every day, but every other day, that it induces these abnormal behavioral changes that persist long after the LSD is discontinued, like for weeks or potentially months after, that are relevant to models of schizophrenia and psychosis with the behaviors. And we've looked at gene expression profiling in the brains of these rats and see a very high significant correlation with genes that have been implicated in bipolar disorder and schizophrenia and psychosis in what's altered by everyday LSD. So timing does seem to make a difference. We don't understand what that is at this point. I know when I was giving LSD every day to my rats, I could tell very quickly that they had become insensitive to the behavioral effects. They also lost a lot of weight. So the theory in sort of the 3 to 4 day dosing is to let the system sort of reset back to normal, let the receptors sensitize, With the every other day, I think that you sort of have these gene expression changes that happen that don't quite get back down to baseline, that then the next 2 days later, so you're sort of bootstrapping up this collection of genes that's more relevant to psychosis than it is to a therapeutic effect. Whether or not that's there in 4-day dosing, nobody knows.
Yeah,
that hasn't been looked at. Lots
of really fascinating and intricate pharmacology that doesn't really fit traditional paradigms.
Yeah, the 5-HT2A receptor is a strange beast. It's not like most other GPCRs or neurotransmitter receptors. It's got some unique features about it and what it does.
Definitely. Thanks for that. Jordan, let me unmute you.
Thanks, Dr. Nichols. Amazing talk. Just a quick question. I'm just wondering if you've looked at neurodegenerative disorders or how you think some of your research might affect those set of disorders, which I know there's attention on right now with whether psychedelics could have effects on Alzheimer's, Parkinson's, things like that. And yeah, just wondering if your thoughts on those.
So we've thought about it. I've thought about it a lot. Looked at animal models. Unfortunately, not enough resources and personnel in the lab to kind of expand off in that direction with the models that we're doing. With Eleusis, the company that I partner with, we've done early stage clinical trials with low dose repeated LSD in a healthy elderly population. Show that there's no negative effects on any physiological or cognitive outcome measure. And that's really in preparation of doing a phase 2 trial in early Alzheimer's patients to potentially be disease modifying in Alzheimer's, not necessarily to treat the depression associated with Alzheimer's, but really be disease modifying. And so that's one of Eleusis' clinical programs, is low dose repeated LSD, essentially like every 4th day, to see if that has protective effects against conversion of MCI into Alzheimer's. So there's movement in that direction, but not in my lab per se. It's more through Eleusis.
Thank you.
Stephanie?
Hi, Dr. Nichols and everyone. Thank you so much for this. It was really interesting and quite a little bit out of my element. But I was surprised to find out about the uveitis because I went through that for a long time and I suffered and it was quite painful, unpleasant to experience. And I actually wonder if separating the whatever science words you used, the psychedelic part from the anti-inflammatory part, if that's actually, like if you're actually taking away part of the medicine. Because I experienced I took a holistic approach to healing and found a new way of seeing while and decided that the painful treatment I was getting wasn't worth it, and I'd rather go through the pain of going blind.
Wow.
And in that process, I actually learned how to see differently and healed. I went back for surgery again, and the doctors said they wouldn't because my eyes had just stopped showing symptoms. And then later, I tried psychedelics. But there's just that magical piece that I wonder if that's actually, and also inflammation in the body shows up with people who tend to have a lot of mental health things.
Right.
So I wonder if taking that away is actually taking away part of the cause. Do you know? Sorry, I'm trying.
No, I know what you're getting at. No, that's of course that is a topic that comes up in a lot of discussions with the potential of taking the psychedelic part out of the molecule. And It's not to say that they're incompatible. In some instances, I think having the holistic approach is very useful, where you treat the mind and the body. And maybe in some instances, for example, you have people who have predisposed to mental illness in their family. They have a first-degree relative with schizophrenia or something like that, where it's contraindicated to take a psychedelic because it may precipitate worsening behaviors.
Yeah.
Or you have your grandma who's really afraid of having a trip, but she'll take a pill. So I think there's really roles for both sides of this, for having psychedelic anti-inflammatories as well as non-psychedelic anti-inflammatories that hit the receptor. The holistic approach or the more targeted approach.
I wonder if you're taking away, or if you're just taking a microdose and you're not getting the psychedelic or the trip experience,
right?
But you're still getting the compound, or I'm not sure.
Well, then again, it's that sort of bypassing the holistic approach, because you're not having the psychedelic effects associated with the microdose. So is a microdose really taking a therapeutic dose of a non-psychedelic psychedelic? And whatever
Say that one more time,
may be. I'm getting caught up in the logic loop here. But with the uveitis, we presented at the ISRP meeting last fall. My colleague Anna Lucis, in a tuberculosis model of uveitis, and it lots of anti-inflammatory effects at the retina and the anterior of the eye, the posterior of the eye. Ultimately, we decided not to go with uveitis because it likely wouldn't be commercially successful because there's just not enough people with uveitis at this point. So we're going, for more generalized retinopathies at this point. But what we did see in our preclinical model data was some pretty significant anti-inflammatory effects in the uveitis model.
Well, that's wonderful. I'm great. I'm so glad to hear that. Thank you.
Yeah, that could be a future off-label use, maybe.
I didn't say that.
Yeah, the data will tell, I guess.
Yeah.
So I think that's a good place to close. We've run over a little bit. So I'd just like to thank Dr. Nichols for being here with us tonight, spending all this time with us and giving such an interesting lecture and bringing, a perspective that we don't hear about a lot. So thank you very much. I will unmute everyone so everyone can give a thank you before you take off.
All right. Thanks for the invitation and sticking around. It was a real pleasure.
Thank you. Great.
Thank you.
Thanks so much.
Thanks a lot.
Good job.
Take care.
All right. Thanks.